Hey everyone! So here we are with lab number 3, our crosses lab. So today, it's a lot of drawing things, not drawing as in like illustrations, but drawing samples from a pool and seeing how that plays out with the genetics. We're going to be working with the genetics of the fruit fly, Drosophila melanogaster. So the lab's going to be broken up into 3 different pieces. We're first going to go back and sit down at the computer and go over the fruit fly manual, so that way y'all pick up on the important components that we need in order to understand fruit fly genetics. Then we're going to come back and take a look at our breeding stations. 3 stations with popsicle sticks that skip the fact that we need to wait 3 weeks for fruit flies to breed. And then after we're done with our fruit fly breeding and Punnett squares, we're going to go over and take a look at our chi-square distributions with random samples. The last thing we're going to do is we're going to tie that chi-square to our fruit fly genetics. So let's go ahead for now and sit at the computer and take a look at our fly manual.
With our fly manual, now taking a look at this electronically. You will of course find the entire fly manual at your disposal for reading on our Canvas page. It's fairly detailed, especially if you want to get into fruit fly biology, which is a huge field of research. But today I'll kind of emphasize some of the pieces that you'll need in order to work in our lab. This lab manual was designed by the Tauber Lab at the University of Leicester in the UK. It's a phenomenal resource that I've seen during my undergraduate studies and is kind of mimicked in other ways by other groups that try to incorporate their research into the work. General table of contents, you see the different components of what a fruit fly is, some of the different mutants, that's what we'll be paying attention to, and also taking a look at the sex of flies. And then they have images of the different mutants to help put a face to the name of the different traits that we'll be talking about in our own Punnett squares. And then we have some protocols. This is stuff that if you want to go into fruit fly biology, if you intend on taking on a research internship at any point in your career, there's no small chance that you will be working with fruit flies. Even though I was a plant biologist, I definitely did some fruit fly crossing both before my undergrad and during my undergraduate career. They're an incredible genetic resource. So the fruit fly, Drosophila melanogaster, is, I won't read this word for word for y'all, but it is a species that travels with humans. So we'll oftentimes see it where humans have spread. This is why you see it in your house. This is why you see it well past some of its original native ranges. They love living in our homes and occupying some of our fruit as it gets older. They cycle very quickly, so they're able to complete their life cycle in less than 2 weeks. This means that they are a great resource in terms of genetic studies in a laboratory environment. You don't have to wait a huge amount of time in order to see the next generation. And this obviously has importance when doing Punnett squares. They have 4 chromosomes, which actually one of my scientific publications was working on gene annotation in the Drosophila dot chromosome. So my career in genetics took me to study the Drosophila genome, which was quite fascinating. We started studying the fly in great detail after Thomas Hunt Morgan, a Nobel Prize winner, really began to untangle some of their genetics, particularly with his discovery of the mutant white eye color phenotype, which is one of the traits that we'll be talking about. The white phenotype, white eye color, is linked to the X chromosome, so it's a sex-linked trait, very interesting. And then, of course, we've actually used the fruit fly to uncover a lot of human genetics as well, as the first alcohol dehydrogenase gene and the study of evolutionary genetics across a wide berth of organisms was first performed in fruit flies. So my first evolutionary biology paper I ever read was a study of DNA substitution rates between human and fruit fly alcohol dehydrogenase. You have a diagram of its life cycle going from an egg to larva to a pupa to an adult and cycling fairly quickly. Even in high school, we were doing some crosses in vials in our trailer classroom, so it was kind of cool, fairly easy to do. Somewhat time intensive, so we won't be doing any of the crosses in our laboratory, but we will be simulating the crosses with our popsicle sticks. There are other things I'd like to move on to rather than spending such a significant chunk of our time performing fruit fly crosses. Now, in terms of sexing the flies, you can identify females and males. So this is actually something that I encourage you to do if you have fruit flies at any point in your home. I have a compost bin at my house, and so we regularly get fruit flies, and I will occasionally capture a couple of them in a jar and then bring them into class and see if folks can sex them. So don't be surprised if I do that at some point this semester as it warms up to help make it interesting for folks who intend to work in any kind of field with insects. So the females have a pointed abdomen with a spike on the top side or dorsal surface near the rear, whereas males usually have a much darker rounded abdomen. Those are really the strongly identifying features. Once you get a dissecting microscope, you can additionally see some structures around their genitalia, particularly whether there's a lot of stuff or a little stuff. Males have more stuff on the ventral surface of their abdomen, and females have the structures, fewer structures externally, more of them are internal. Males also have sex combs, so a row of short, thick bristles that appear as a dark mass on the fourth segment of the front leg. So you see them on the legs of the fly, and they're fairly remarkable. Like, you'll definitely notice them. Here we have an image of a female fly with a lighter colored abdomen and a male fly with a darker colored abdomen, a much more rounded abdomen, whereas the females comes to a point. The pigmentation is really going to be your best bet, though, of course, there are some mutants for pigmentation that will make it more difficult compared to the wild-type fruit fly. Note, these are, of course, not images shown to scale. A fruit fly is incredibly small. I don't remember the actual centimeter measurements here, but we're talking about an organism that is quite small. Here we have an image of the male and female in terms of a head-on view for the sex comb. So you'll see the sex combs here on the front forelimbs of the male fruit fly. You additionally see more of the external structures, so the copulating organs for the fruit fly, and then comparing that to the lighter colored abdomen without any external structures of the female. Now, in terms of our mutants, and we'll be having some of these in the Punnett squares today during lab, and we will additionally, potentially, be having some of these during the actual exam or future Punnett squares. There's lots to be practiced with Punnett squares, and so I just want to give some idea of an appearance of these traits in the fruit flies. White eye color is quite noticeable. You'll see that the wild-type fruit flies had the very distinct red eye color. You will occasionally see, even out here in my compost pile, I've spotted a couple white-eyed fruit flies. It's kind of cool. They, again, have white eyes. It's a mutation away from the normal red-type eyes. We have a curly-winged gene, so a gene for curly wings, a gene for a yellow body. This, again, is one of the traits that that can be appearing in any of the Punnett squares during the class. So you'll want to note which genes are sex-linked on the X chromosome versus which genes are autosomal, not on sex chromosomes, as they will give you a skew in terms of the gender. The ebony fly is something that I've personally found a little bit more difficult to identify male versus female because it does have a lot more dark pigmentation, though if you have a quite bright light, it does help elucidate that difference. It is an autosomal gene. The eyes absent gene is actually something that I have not seen firsthand in any of my fly research, and as you'd expect, the phenotype is that their eyes are missing. This is an autosomal gene. The Dihybrid are a trait in which their wings are extended above and away from their body. This is very different from a lot of the insect phenotypes, so it's quite notable. Of course, we also have the vestigial winged phenotype. This is one in which the wings do not fully develop. So you can imagine that there would be very small wings here. So that's kind of it in terms of what you need in order to succeed today in lab. I'd of course encourage you to skim through the remainder of the document in case you ever want to work with fruit flies. You can put them to sleep. We have a fly nap, and I'd encourage you, if this is something that you'd like to do for your career, it's very easy for us to harvest some fruit flies from someone's compost pile, and you can practice anesthetizing them at any point during our semester, and seeing if you can sex them and identify any potential mutant traits. You additionally can see if you want to propagate them, so do some fly crosses. We have the materials to make the solution as well as the vials, and so you are welcome to, but it's not something that we'll be doing in our lab. You're welcome to organize your own fruit fly crosses. So with this as our background and hopefully a good understanding of what the fruit fly looks like, so now, since we'll be talking a lot about its genetics in our Punnett squares, we can go ahead and I'll see y'all back in the classroom where we'll talk about fruit fly genetics in terms of Punnett squares and popsicle sticks.
All right, so now that we have a good idea of what fruit flies are as not cute but useful organisms, we have our 4 stations for our fruit fly crosses. Now we're using these stations rather than trying to breed fruit flies over the course of several weeks. You could do that in perhaps in other biology classes, you may have, but in interest of time, we're going to stick to these 4 popsicle stick stations. So the way to sample, the way to accelerate that breeding process is to go to each one of these stations. They should be mixed up. And then you'll pull out 10 popsicle sticks. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Now you're going to want to grab these real quick and jot down the genotypes that you see on, or the phenotypes that you see on here. Because with each of these popsicle sticks, you can see what the fruit fly looks like, but you cannot tell what its genetics are. So looking at this sample from station 1, what we are looking at, and again, this is in somewhat in combination with the table from the lab. So you can see the table itself, it gives you a little bit of a key. We can see that our fruit flies had wings that were VG or vestigial, and it was a male. So you can see the gender on the right written in red, and what the fruit fly looks like on the left. A plus indicates wild type. So now what we're going to need to do for each one of these sets is count out how many males and females we get and how many vestigial and wild type we get. This is going to be our pool for this set. Then I'm going to go through and collect 10 popsicle sticks for each of them. And then because the class needs to operate with replacement, you will take the popsicle sticks and put them back. If you're doing this lab completely virtually, we will go ahead and show you the statistics or the counts for the progeny after I go ahead and pull out 10 popsicle sticks from each station. For now, I will send y'all up to Becca at the whiteboard, who'll be going over the basic structure of each of the Punnett squares, because we're going to need to correlate our counts of phenotypes with genetic Punnett squares. So head on over there.
All right, welcome to the whiteboard. We came up here to look at the difference between monohybrid and dihybrid crosses. So we'll start with our monohybrid. This is when you only have one allele. So we'll call it A. We've got A1, we've got A0. Or excuse me,
One.
we have
One gene.
one gene with 2 alleles. Sorry. So A1, A0. We're going to cross this with another A1, that's an A. Another A1 and an A0. These guys are both heterozygous. So when we cross these, all that we're going to need is a 2x2 or a 4 square Punnett square. And we'll put them both up here. A1, A0, A1, A0.
Does gender matter? Do you know whether male's on top or female's on side? Does it matter, Becca?
Right here, it doesn't make any difference. all you're looking at is the gene.
Perfect. How does the monohybrid cross differ from a dihybrid?
So a dihybrid is where you're looking at two different genes that have 4 alleles because there's 2 apiece. So let's say it's A1, A0, and then B1, B0. So same thing, we'll cross it with one just like it. A1, A0, B1, B0. Now in this case, since there are 4 different alleles here, we need 4 squares along the top and 4 along the bottom. So this will be a 16 box square.
Now, how many letters are going to go in each column? Is it going to be 2 A's per column and 2 B's per column, or are you going to do 1 A and 1 B per column?
Well, to figure that out, we're going to use a method called foiling. So the easiest way to do this is FOIL. First, all right, now that Paul picked the camera back up, we have FOIL. So first, now O, we have the outers, I is inner, and L is last.
Okay, so those are our pairs. We're going to be pairing 1A with 1B within each genotype of each parent. So above each column, you should have 1A and 1B, and you should look at the alleles based on the foiling method. Very neat, Becca. And now what are you going to expect to see on the other side?
Now down this side, it'll be the exact same thing with your A1B1, A1B0, A0B1, A0B0, because these are both the same.
Perfect, and that works for a dihybrid. Now you won't always be getting dihybrids today in class, but it's a good, useful blueprint to look at with these 2 different Punnett squares, monohybrids here on the left, dihybrids on the right, as we look at variance between our traits in our different stations. Now the last thing, Becca, is we got a couple of vocab terms that I wanted you to cover before we look at our station counts.
Yes, so things you need to remember is there are genes that are dominant and there are some that are recessive. Dominant means that it completely masks. You're not going to see the recessive at all. If it's recessive, that means there are no dominant, because if there was a dominant, you wouldn't see the recessive. Then there's sex-linked. So that means it could be held on the X chromosome. Now, males don't have 2 X chromosomes. So that's why sometimes sex-linked will be skewed because of those X's or missing X's. Then there's linked, which means that the genes could be on the same chromosome or on different chromosomes. Next, there's additive, which means how much of something there is. And finally, true breeding, which means that it is the same. So like we have our A1, A0 here. True breeding would mean that you have an A1 and an A1, not that so they would be homozygous rather than heterozygous.
Perfect. All right, let's go ahead and take a look at some of the data from our stations. All right, so I finished out laying out all the popsicle sticks and our counts. So let's take a look at our data and then how we summarized it. So for station 1, we see that we pulled some vestigial winged females and wild-typed winged males. Now, looking back at the sheet, we know that wings are not sex-linked. So here we don't really need to track the gender on the right side of the popsicle stick, but you can keep track of the fact that we see 1, 2, 3, 4 vestigial, and 1, 2, 3, 4, 5, 6 wild type. So that's our data from station 1. Now we can go ahead and take these popsicle sticks back for another group to use. For station 2, again with a monohybrid, so 1 trait, we see wild type wings, wild type wings, wild type wings, and vestigial wings, 3 of them. So we see 7 wild types and 3 vestigials, again, not sex-linked. Let's go ahead and put this one back. Mix them up again. For station 3, we now have a dihybrid. So that means that we're going to have 4 categories. So we didn't separate these out. You can see here, and I'd recommend pausing the video if you want to count these out yourself, but you can see where the wild type wings and yellow bodies are, or you can look at our summary table where we see how many vestigial wings with wild type bodies, vestigial wings with yellow bodies, wild type wings with yellow bodies, a lot of those, and wild type wings with wild type bodies. Go ahead and put those back in. For our 4th and final station, our station with sex linkage. So now you do have to keep track of gender here. We see males with white eyes, and just so happened that this first pull was 1 male with 9 females. That's a quite strong gender skew. But we can see the statistics for it gathered here. 1 male with white eyes, 4 females with white eyes, and 5 females with red eyes. Now, just because that gender skew is pretty strong, we pulled again. And this time, we got 6 males with 4 females. And we wrote down the statistics on the back of this page, where you can see 2 white-eyed males, 4 red-eyed males, 1 white-eyed female, and 3 red-eyed females. And now with these statistics, let's go ahead up to the board and draw what we think our crosses were for each of these.
We are back at the whiteboard. We're going to look over what we have, like what we're given, to see if we can figure out what the genotype of the parents was, based off of the phenotype and our counts that we got. So we are told this is a monohybrid cross with the parental generation being a normal wing male and a vestigial wing female. So it says the male is not true breeding though. So we know that our male must have been a wild type and a vestigial, because he's not true breeding, so he's heterozygous. Meaning, if he showed normal type, this one is dominant. Ooh, my marker work here. And then we can get our female, and it says we have a vestigial wing female. So, if this one's recessive, we have to have 2 of these. So then we have wings vestigial and wings vestigial.
Perfect. Great explanation, Becca. I like that.
And do our little cross here. We get a wild and a vestigial, and a vestigial, wild vestigial, and vestigial.
So what are our expected phenotypic ratios from our progeny?
The expected from this is going to be 50-50. This one would end up being wild type, because that's the dominant. These 2 would end up being vestigial.
Cool. And so we expect 50-50, and we got 40-60. Is that close enough?
It's close enough to have only 10 of the offspring.
Perfect. So we are looking for whether these genotypes make it possible to produce those phenotypes in the offspring. So that sounds right by me. Looks like you have the genetics mapped out here. Let's take a look at station 2. I'll be doing that one. So let's switch spots. All right, so here we are with station 2, where we saw 3 vestigial and 7 wild-type winged offspring. Now the difference in comparing station 2 to station 1 is that this is going to be our F2 generation. So rather than looking at the progeny directly of our parents, we need to take our parental generation, produce an F1, mate within our F1 to generate our F2s. So what information are we given about this station? Well, it says that the parent generation is a normal wings male cross with vestigial wings female, with both being true breeding. So in our parental generation, wings male, wild type, wings male, wild type, crossing a female with wings vestigial, wings vestigial. This is going to make all 4 of the same offspring with a wild type allele and a vestigial allele. So we know that the F1s are all heterozygotes. What do we get when we cross a heterozygote and a heterozygote? So let's draw out this again. Heterozygous male crossing a heterozygous female. This plus this get put into this box. Wild type, homozygous wild type. This plus this get put into this box, giving you a wild type vestigial. Vestigial plus wild type, top right corner. And it doesn't matter which order you list them, they're separate alleles. So I'm listing them with wild types first, vestigial second. And then our last box, vestigial plus vestigial. We know that this is not a sex-linked trait, as we've established previously, so we don't need to track gender in this. Looking at this mess of a Punnett square, we can go back and count how many wild type were made. 1 of these. Wild type vestigial. 1, 2, and then vestigial would be 1. But because we know that wild type wings are dominant, we can lump these together for a ratio of 3 to 1. 3 wild types per 1 vestigial. That's based on these genetics. Let's take a look at our offspring. We get a ratio of 3 vestigial to 7 wild type. That's a ratio of roughly, if it was 3 to 1, we'd expect 7.5 to 2.5. As of yet, I have yet to find half a baby or 0.5 of a baby. That doesn't sound like something that biology is going to be able to deal with. So 7 to 3 is close to 7.5 to 2.5. So yeah, I believe this true breeding vestigial versus wild type in our parental generation would create heterozygotes in our F1, and we are indeed generating this ratio of vestigial to wild type in our F2s. So hopefully that Punnett square makes sense, and you're looking at different generations. Let's now hand over the reins to Becca to take a look at our dihybrid cross. And the thing that I want y'all to keep in mind when Becca's going over the Punnett square is that thus far we've seen the wild type be dominant to the mutant, the vestigial. Scrub that from your brain. We know that wild type is dominant in wings to vestigial, but we can make no assumptions about whether wild type is dominant to any of the body colors at another locus, another gene. So come on over, Becca.
And here, let's look onto our section 3. So, what we need to recognize on here, again, we're looking at F2 ratios. So this is not just from our parental generation. But we do know what the phenotypes of the parental generation were. So with that, we can distinguish their genotypes since we figured out what's happening. What you need to know is that these are true breeding. Well, it doesn't say that in the packet. Make sure you know that because they are.
That would have been announced in class, so now that we're pretending this is class, the Station 3 parental generation is true breeding. Take it from here, Becca.
All right, so we know that our males are normal winged, normal bodied. If they're true breeding, that means we have wings wild type, body wild type, and body wild type. Easy enough. Now we'll look on to our females. They tell us that the females are vestigial, yellow body. So, again, true breeding, wings, vestigial, wings, vestigial, body I for yellow, body I for yellow. Now, what we'll need to do is cross these in order to get to our F1 generation. So, what we could do here is make a 4x4 dihybrid Punnett square, and it'd be this really big thing, and it would take a lot of time. If you need to, go ahead and do that. Otherwise, we're getting to the point now where we can use some shortcuts. So, since we know that these are homozygous for these traits, we can look at this and determine that it is going to be wings, wild type, body, wild type. That's all you're going to get from it. Wings, so wild type, outer, wild type, inner, and last. They're all going to be the same. Same thing here. This one's homozygous as well. So wings, vestigial, body, yellow. These guys are what we're crossing. No matter how you cross them, you're going to come up with the same things. You're going to have a wings wild, wings vestigial, one of each. You're going to have a body wild and a body yellow, one of each.
So it looks like the F1 is fully heterozygous.
Mm-hmm.
Perfect. So now for our F2s, we won't be able to do the shortcut here. So let's go ahead and draw out a 4x4 Punnett square and do the foiling for that. You will note how much neater Becca's handwriting is than mine, so thank you, Becca.
I try. One of us has to.
Yeah, right?
All right. Now, since this is all of our F1, we need to figure out what the different pairs are. So what we're going to write up here and along here, how we're going to make this. So we're going to do our foiling again. We're going to have, let's see, first, outer, inner, and last. That's what we got.
Perfect. So how many different sets of alleles do we have?
4.
Cool. Pairing each of our...
Broke up a little funny, but...
Perfect. No, that's great. So we see wild type wings with wild type body, wild type wings with yellow body, vestigial wings with wild type body, and vestigial wings with yellow body. And now what do we know about which traits are dominant versus recessive, Becca? Does the station give us any other clues about that?
Yes. So the second thing we need to look at here, it tells us the resulting F1 is entirely wild type for wings and yellow bodied, with equal female, cross, equal male. So since male and female are equal, it's not sex linked, don't need to worry about it. But they say that they're wild type for wings and yellow bodied. So wings, we already knew that. Wild type, that's what you get. But if they're yellow bodied, even with, so this is what we're looking at here, wild type wings, if they're yellow bodied, that means yellow has to be dominant, not the wild type.
Perfect. So that was that little foreshadowing that I did before I stepped behind the camera. Becca guided us through that very well with wild type wings being dominant, but the non-intuitive yellow body being dominant. So let's take some time and draw us out that Punnett square and see what our progeny ratios are. Go get them.
All right, stick with me here. I'll write this all out. You guys can too. And it'll be the same thing on both sides.
Perfect. And now for our sanity here, Becca, we could go ahead and go through and put all of the genotypes in here, but let's do a little shortcut. Since we're only looking at phenotypes, let's go ahead and put what is the expected phenotype in this box.
Okay. So since we're looking at wild types, all that we're going to get is, let's see, how do we have these written?
Wild type wings.
We're gonna get wild type.
Yep. So and wild type wings and wild type body. Perfect. There we go. Thank you.
Yep. So now we have wild type with wings, that's all we got. And bodies, we have a yellow body. So that one's gonna get it.
Perfect. One yellow body allele is sufficient to make that entire locus yellow body. It's the dominant one.
And here we have a vestigial, but we have a wild type. That's our dominant. And here we only have wild types. Wings again, wild type. Body yellow.
Perfect.
Wings, wilds. Body. Got our eye here. Body yellow. Okay.
Halfway done.
Whole row's the same there.
Perfect. Yeah, the whole row's the same there because one of the progeny has both dominant alleles, so it's going to create all dominant offspring for both loci. Those are the kind of things that you will learn to observe or train yourself to observe as you do more of these Punnett squares. And y'all should definitely be doing a lot of Punnett squares.
Lots of Punnett squares. Yellow. All right.
Perfect. Let's do some counting. So how many, since we know that yellow is dominant and wild type wings are dominant, let's go ahead and count how many squares have wild type wings and yellow bodies. Nice change of color. Perfect.
All right. We want wild type wings and yellow bodies. There's one. That one's not.
2,
One is.
3, 4, 5, 6, 7, 8, 9. Okay, so that's underneath the Punnett square. Go ahead and write that. 9 wild type wings with yellow bodies. Let's change from a star to a triangle or some other shape or color. Oh, we got colors.
We have colors.
Pick the next one that we're going to go over.
Let's go with our wild types.
Okay. 1, 2, 3 of those. Perfect.
All right, 3 of those. Now, what we got left? We've got vestigial and wild type.
Just the one.
That's it.
And last but not least,
Let me spread back again in this corner. And last one, we've got our vestigial and our yellow bodies. Let's see, circles. 3 of those.
all right, so this produces your familiar ratio of 9 to 3 to 3 to 1, where you have the 2 dominant loci, the 2 dominant alleles rather, being a 9, a dominant mixed with a recessive, a 3, a recessive mixed with a dominant, a 3, and the recessive at a 1. Now, if you're expecting a 9 to 3 to 3 to 1, and we got a 7 to 2 to 1 to 0, That seem believable?
It's pretty close.
Perfect. So it is possible. Your ratios in class might be a little bit different, but we definitely believe that these genetics could have produced those phenotype ratios in the progeny. Thank you, Becca, for that Herculean task. Dihybrids on the whiteboard are never easy. I'll now take over because we're going back to single trait, even though they're sex-linked. All right, so here we are with our fourth station where we got our 2 sets of data for, because, you know, the first time random sampling, we only pulled 1 male, 9 females. Now, what do we know about this? First and foremost, from our fly manual, you know that eye color is sex-linked. You also know that white eyes are a mutation. And we know that fruit fly gender, males are XY, and Y has no allele for eye color, and females are XX. So males are going to be hemizygous, only 1 allele. Now it says that our parental generation, the male is white-eyed. So let's go ahead and use an XW, Y. I should write this bigger. XW, Y. The female is red-eyed, but we don't know whether she's true breeding or not. So the female can be red-eyed, or the female can be red-eyed, white-eyed. If red eyes is dominant, this would work. If red eyes is not dominant, this would not work. We're going to have to look at our progeny ratios in order to try to figure this out. We don't know anything about the F1s other than these are their ratios. So, if we make a Punnett square, where we have the male on the top, X with a W, Y, and then let's go ahead and make a female that's RR and see what progeny ratios come out. XR, XW, XR, XW, XRY, and XRY. What this means, assuming that R is dominant, is that all of the females would have red eyes, and all of the males would also have red eyes. Means we don't see any white eyes. So, do we actually see that in our progeny ratios? No, we're seeing some white-eyed females. So that means it's unlikely that our female is homozygous. Let's instead do this cross with XR, XW for our female, and same genotype for the male of an XWY. We would make a XR, XW, 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 XR, Y, XW, Y. That means we should see red males, white males, red females, white females. Well, here I didn't get a lot of males, so that makes this a little bit more difficult. But here I did, right? I got both white males and red males, and white females and red females. Now, they're not perfectly in a ratio of 1 to 1 to 1 to 1, 2 to 4 to 1 to 3, but that's random chance for you. This is the Punnett square that we should be looking at. Red dominant to white, and your female should be heterozygous. Now with all of our Punnett squares complete, accounting for all of our data, let's go ahead and take a look at our chi-squareds. Some random sampling and a little bit more math. All right, to the last section of the lab. I know it's been a long one. We're taking a look at chi-squareds. We're going to be revisiting this again in lecture, so please make sure that you can supplement this material with the posted lecture. But for now, the basic idea behind a chi-squared is to tell whether it is likely that you would get some observed frequencies, like we were looking at our progeny ratios before, those observed frequencies, based on what you would expect given the genotypes of their parents. But before we get there, we're going to be a little bit more simple. Now, of course, in the era of COVID, the lab asks you to look at M&Ms and eat those delicious M&Ms, the different colored M&Ms, but we unfortunately cannot do that. So instead, I got a bucket of markers. Now, in theory, what I've done is I've taken two sets of markers, reds and purples, equal numbers, and throw them into this bucket. I want to draw randomly from this bucket and see whether the draw that I pull is likely to occur from a bucket that started out with even numbers of red and purple. Now, if it was skewed some different way, like a linkage problem, there are ways to test that, and the lecture will go over that. But for now, we're assuming a 50-50. So we want to set up our problem. In order to do so, we need to see how many red markers and how many purple markers we observe versus how many we're expecting. Now I am going to make sure to pull 5 markers. So the total number that we expect is 5. The total number that we observe is 5. Now if there's even numbers of red and purple and fractions are allowed, what is your expected number of reds? Well, half of 5 is 2.5. Half of 5 is 2.5. And the chi-squared equation is the sum of our observed minus our expected squared divided by expected. And we're going to be summing over each of the terms. So we're going to be summing over red and purple. We're going to need to have this term derived for red, this term derived for purple, and then add them together. So let's go ahead and get our observed counts here. Try not to forget. 1, 2, 3, 4. Oh boy. So that's 4 purples, 1 red. 4 purples, 1 red. Let's set this term up. So now, making this equal, we'll do reds first. We will have 1 observed minus 4 expected squared divided by, oh, my bad, y'all. 1 observed minus 2.5 expected divided by 2.5 expected. You're going to add this to our purple. 4 observed minus 2.5 expected squared divided by 2.5 expected. Now, I can't do the math in my head. So we've got 1.5 squared, and this will be 1.5 squared. We'll be right back with the calculator to make sure we get that. So sorry about that. Forgot the fact that we needed a calculator. 1.5 squared, 1.5 times 1.5 gets you 2.25. Divided by 2.5 is equal to 0.9. It's going to be the same thing over here. 1.5 squared divided by 2.5 gets you 0.9. 0.9 plus 0.9 is equal to 1.8. So our chi-squared statistic for this, with a pool of only 5, and that's going to be the important fact here, is 1.8. Now from the lecture, we know that our chi-squared threshold for a 95% confidence interval is any value above 3.84 means that this bucket did not start out 50-50. So even though we pulled 1 red and 4 purple, it is likely to happen in a 50-50 bucket because we only had 5 pulls. I'd encourage y'all, you can do one of these at home. If you only have a small number of pulls, like 5 pulls, you can deal with, or the chi-squared statistic is robust to still not be able to reject the hypothesis. Try doubling these numbers. Let's say that instead of getting 5 pulls, we got 10 pulls, and we got 1 red and 9 purples. What would our chi-squared statistic look like there? So I'd encourage you to, whether you're at home or whether you're in the classroom, set up a 50-50 bucket, and honest to goodness, surprise yourself. Not gonna lie, I was surprised to see 4 and 1. The remainder of the lab is a thought exploration for how small sample sizes and chi-squared relate. I'd recommend looking additionally at the lecture on chi-squared to see how this might connect to linked and unlinked loci, and then talking about how your distances and centimorgans might vary based on whether you're really close to the 50-50 distribution, 50-50 being unlinked loci, with a loci that's, or 2 sets of loci that are 48 map units apart or 48 centimorgans apart. That said, please spend some time exploring some chi-squareds. Please make sure that you understand our different Punnett squares. These topics are reinforced both in lecture and in lab. They go hand in hand. There's not a lot of distinguishing between the two for these 2 weeks. So go back through your lecture notes, see what else you need in order to complete this lab. I will see y'all next week.